Hello class, this is Dr. Lyons, and in this chapter we're going to be talking about DNA technology. So this obviously isn't a picture of DNA technology, but it is a picture of a lionfish, which I took. So sorry if you were expecting a picture of DNA technology. So in this chapter, we're going to mostly be focusing on applications of DNA technology. Right, so before we go on, I want you to think about some potential applications that we might use for DNA technology. So pause the video, think about that for a second, and then go on. So one of the really important places where DNA technology can be applied is to pharmaceuticals and the medical field in general. So for instance, something that pharmaceutical companies have been using for years is something known as recombinant DNA technology. So this is actually how a lot of therapeutic hormones, such as insulin and youth growth hormone, are made. And essentially how it works is we trick bacteria into making proteins that we need. So back before this technology existed, people that were diabetic for instance, that needed insulin, they would take insulin from other animals. Essentially, they were taking insulin that was raised in livestock, essentially. Whereas now all of the insulin that diabetics take is made with this technology. And how this essentially works is you take a bacteria. And you take a plasmid out of that bacteria, which is essentially a little string of DNA. And what we do is we insert a particular gene into that plasmid. In the case of insulin, we would insert the insulin gene into that plasmid. Because remember, back in chapter 10, we talked about how we start with DNA. We start with a gene of DNA, say the insulin gene. That insulin gene existing in the DNA, then through the process of transcription, gets made into an insulin gene in the format of RNA. Then that RNA leaves the nucleus, goes away from the DNA, and goes into a ribosome outside of the nucleus, where then in the ribosome translation takes place. In translation, that's where we take those letters of RNA, like the A's and U's and C's and G's, and we convert them into amino acids. When we put a bunch of amino acids together, we get a protein. So essentially what happens in our bodies is we take our insulin gene, our cells then take that information in that gene and use it in order to make the insulin protein, which our bodies need for breaking down food. What you're doing here is we're taking that particular gene and splicing it into a plasmid, so into a stretch of DNA, and putting that DNA back into the bacteria. Right? So this bacteria now has this little insulin gene inside of it. And when that bacteria then reproduces, it is making more and more bacteria that has that DNA inside of it. And what that bacteria is going to do with that DNA, with that gene, is it is actually going to use transcription and translation on it. So it is going to make a whole bunch of insulin, of human insulin, using that DNA. So essentially we've tricked this bacteria into making something that we want. In this case, tricking it into making proteins. So we're using it to our advantage. So anybody that you know that has ever taken insulin, this is how that insulin is now being made. Is by tricking bacteria into making those proteins that we want, proteins such as insulin. So that's one really important application. Another really important application is forensics, right? So forensics now very much relies on DNA technology. Pretty much since the O.J. Simpson trial in the early 1990s, most, well, not necessarily most, but a lot of court cases where a lot of court cases where there are really malicious type crimes oftentimes use DNA technology in order to pinpoint a particular suspect at the scene of the crime. And so there's a couple of important DNA technologies that are used in forensics. One is what is known as polymerase chain reaction. So you might recognize that word polymerase. We talked about it in the context of DNA polymerase and in the case of RNA polymerase. So what DNA polymerase does is that is a protein that makes DNA. It makes copies of DNA. It makes more and more DNA. We talked about that back at the beginning of chapter 10. So the polymerase chain reaction essentially does that. It takes a small amount of DNA, so say a particular small sample of DNA that was left behind at a crime scene, and then it replicates it. Because, typically, people that commit crimes, they don't go out of their way to leave a huge amount of their DNA behind, right? In fact, a good criminal probably, well, not an evil, but efficient criminal is going to try to leave as little DNA behind as possible. But even if they leave a very small amount of, say, blood or skin cells or hair, with polymerase chain reaction, you can take the DNA that is inside that very small sample and then replicate it. So you have more DNA to work with to do other such as short tandem repeat analysis. So what this process is and what this technology is, it's essentially a way of matching DNA from a sample with an individual. So what an actual short tandem repeat is a section of DNA that's repeated over and over and over again. And we have many of these short tandem repeat areas within our genome. There's lots of places where you might have, say, AGAT, then another AGAT, 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 over and over and over again. So we have these different repetitions of say, that AGAT. And from individual to individual, there's a lot of variation in how many times those are repeated. So for instance, for a short tandem repeat site, I might have 1, 2, 3, 4, 5, 6, 7 repetitions of AGAT. Another person might have 8. Another person might have 9. Another person might have 100. So it's very variable between different humans. And when you then use one short tandem repeat site and take information from then another site and another site and another site, you can essentially make a genetic barcode of one individual person. And with that barcode, then you can see if it matches, say, the DNA at the scene of a crime. So in this case, say we've got crime scene DNA. And at short tandem repeat site 1, there are 1, 2, 3, 4, 5, 6, 7, 7 different repetitions there. And in site 2, we have 8 different repetitions. What we see in the DNA of a suspect is that it matches for one of those sites, but it doesn't match for the other site, meaning that the DNA that was found at the crime scene would not match this particular suspect's DNA. So they would probably rule out that suspect as being a person that might have committed the crime. So that's what short tandem repeat analysis is. It's a way of matching very precisely DNA from a crime scene to a particular suspect. Okay, so another potential or another DNA application that short tandem repeat analysis can be used for is parentage analysis. Right, so we all know about shows such as this, like Maury or what's his name, Springer. All those different types of shows where they sensationalize people figuring out whether or not they're the father of a particular baby. So of course these shows are garbage, but in the case of short tandem repeat analysis, it's actually a very precise way to determine parentage. Because parents will have very similar short tandem repeats to their offspring, right? So in cases of parentage, oftentimes short tandem repeat analysis is what is actually done. Another kind of interesting application of DNA technology is figuring out what happened in the past. So looking at history conundrums, for instance. Right, so I'm assuming most of you recognize the guy on the left, Thomas Jefferson, right, one of the founding fathers of the United States. Probably fewer of you know who Sally Hemings was. So Sally Hemings was a slave of Thomas Jefferson, and for many, many years, historians speculated that some of the children that Sally Hemings had were those of Thomas Jefferson. Essentially, there'd been this long-standing thought that the two of them had had an affair with each other and had produced some babies. Thomas Jefferson was married, so obviously no bueno. And Sally Hemings was his slave, so there was a lot of like bad power dynamic going on there. So it's a pretty contentious thing because of course he is a founding father, but this is a pretty bad thing that he might have done. So essentially the question is, did they have children? And in 1998, scientists looked at short tandem repeats within the Y chromosomes of Thomas Jefferson's uncle's descendants. So why they did that, why they used Thomas Jefferson's uncle's descendants, is because Thomas Jefferson would have the same Y chromosome as his uncle, because Y chromosomes are passed on from father to son, meaning that Thomas Jefferson's dad would have had the same Y chromosome as Thomas Jefferson's uncle, meaning that Thomas Jefferson would have the same Y chromosome as his uncle. And in the case of Thomas Jefferson, his lineage stopped. So there are no living male descendants of Thomas Jefferson that we could test. But Thomas Jefferson's uncle does still have descendants that are alive today. So essentially what they did was they looked at the Y chromosome of Thomas Jefferson's uncle's descendants, so people that are still alive today, and they looked at the descendants of Sally Hemings that are still alive today, and they found a match. So they found that indeed some of the offspring that Sally Hemings had were fathered by Thomas Jefferson. So in fact, this really dark part of U.S. history is completely true. Thomas Jefferson did in fact have children with Sally Hemings. We can also look at broader human history with DNA technology. So for instance, we can do what's known as DNA sequencing. So that is looking at sections of DNA. So rather than just reading just short repeated areas, short tandem repeats, DNA sequencing is looking at whole stretches of DNA and reading all of the A's and the T's and the C's and the G's. Genomics takes that one step further. So that is reading all of the DNA in an organism. So every single little bit of DNA in an organism. And this is a field that has really, really come along a long ways in a very short time. So the first animal to have its genome completely, read is that of us humans. So in the mid-90s, the Human Genome Project was started. It was finally finished in 2003. So something about like 17 years ago or so, the human genome was finished. And it cost millions and millions of dollars, took almost 10 years to finish. Whereas now you can read a whole human genome for something like $1,000, and it only takes a couple of weeks. So the technology has gotten way, way better for reading genomes. Something kind of interesting that came about from looking at genomes is comparing ourselves to our sister species. So in 2013, the Neanderthal genome was finished. Neanderthals were another species of human that were occurring in Europe, and there are some really good fossils of Neanderthals in caves in some of the colder parts of Europe. And so scientists in 2013, they went and got the DNA from those Neanderthals in those caves, like frozen in those caves, and they read the whole genome of it. And what they revealed is that there is some interesting similarities between the genome of humans, of us, and of Neanderthals. Essentially what they reveal happened is that when humans, our species, Homo sapiens, when our species went into Europe, where the Neanderthals were, we essentially started interbreeding with them. So people of European descent tend to have a little bit of Neanderthal DNA inside of them. And most of us have at least a little bit of European ancestry in us, meaning that most humans have a little bit of Neanderthal DNA inside of them. So that's what the very interesting thing that was revealed in 2013 is essentially when you look at us humans, we're kind of two species in a way, because we are mostly Homo sapiens, but we also have a little bit of DNA from Neanderthals as well. So we're kind of two species in a way. So this kind of shows a map of what I was kind of just getting at, right? So here's Europe, of course. The black lines indicate waves of our species moving around in different, in how long ago that occurred BP before present, right? So our species originated in Eastern Africa. When we, start spreading outside of Africa, there were already Neanderthals here. There's another species here known as Homo erectus. And essentially, our ancestors, our ancestral Homo sapiens, started interbreeding with them. So humans were interbreeding with Neanderthals here, and now there's very good evidence that humans were interbreeding with that other species over here. So in fact, our species is kind of like three species, right? So we're mainly Homo sapiens, but we have some DNA from Neanderthals, and we have some DNA from a species known as Homo erectus that occurred in Asia a long time ago. Okay, so another really important application of DNA technology, and one that is highly contentious, is what's known as genetically modified organisms. Right, so these are organisms that we have inserted genes into or modified the genes of, right? So we have changed them around to be useful to us. So in fact, if you think all the way back to the first technology we talked about, recombinant DNA technology, that is essentially making a genetically modified organism, right? So, so those bacteria that we talked about that we can trick into making insulin, they are essentially genetically modified organisms because we have changed them around. However, now we can do this with a lot more things than just bacteria. And in fact, you might be wondering to yourself, have you eaten a GMO? Well, for all of us living in the United States, the answer is definitely yes. So in the US, better than 80% of all the corn that is produced is genetically modified, and better than 90% of all the soy that is produced is genetically modified. Right, so we are all consuming lots and lots of genetically modified organisms. So why you might genetically modify something is to make it easier to grow, essentially. So for instance, there is a type of corn known as Bt corn, and that corn has a built-in insecticide. Right, so instead of a farmer having to spray insecticide on that corn, now that corn has a built-in insecticide into it. So it's less likely to get eaten by pests. There are strawberries that have been made that have antifreeze built into their DNA. Essentially, this makes it such that if there is an early frost, it won't kill all of the strawberry and make it, and destroy all of the crop. So that's why you would do that. There is a type of rice that has extra beta carotene put into it. Right, so it's essentially GMOs that are in our grocery stores are essentially there because they're easier to be made, or there are things about them that make them more nutritious. So you might be wondering, okay, those were all plants, but are there any animals that are genetically modified? The answer to that is yes. So a good example of this is what's known as the AquaAdvantage salmon. So this is a salmon that is an Atlantic salmon, so one species, Atlantic salmon, but they have put into it growth genes from a Pacific salmon. So we've essentially taken one species and spliced in genes from a different species. And why you would do that is kind of shown at this picture at the bottom. Right, so a normal Atlantic salmon, when it's 1.5 years old, it's about 13 inches and about 3 pounds. Whereas if you take this Atlantic salmon and put those growth genes from Pacific salmon into them, then you have a fish that is this big at 1.5 years. So it weighs twice as much and it's almost twice as long, right? So it makes a much bigger fish. And so for a farm that is growing salmon, so a farm that is doing aquaculture salmon, it's going to be much better for them to grow this AquaAdvantage salmon because they get bigger faster, and so they can make more money. And so this is something that was approved by the FDA. But it hasn't reached the market yet because right before President Obama left office, he put in an executive order to stop this so that the FDA would essentially have to do more testing on it. It is, however, on the market in Canada. So we can, over these years, we can take a look at what happens to Canadians when they eat it. They can be our little test dummies up there in the north eating AquaAdvantage salmon, and we can see what happens to them. And then eventually, someday, I assume this probably will make it to shelves in the US. Okay, so one last DNA technology that I want to talk about is CRISPR gene editing. This is a very, very new technique. It was only just built just a couple years ago. And what it essentially does is it is a very new, precise way for exchanging or deleting genes in virtually any organism. And so what CRISPR actually refers to, what that acronym refers to, is the immune system of a bacteria that bacteria uses for fighting viral infections. So how viruses work, including COVID-19, is that viruses essentially insert their DNA into cells, and those cells then start making more viruses using the DNA from the viruses. And then those cells, they start then using that DNA and making more and more viruses, right? So that's what is happening inside of anybody that has COVID-19 right now. The COVID DNA is the COVID virus is inserting its DNA into the cells of that person, and in those cells are then making more COVID-19. So how bacteria fight this is they have this CRISPR system where they have these proteins inside of them that will look for foreign DNA that is inside of their genome. And when it finds that foreign DNA, it cuts it out and removes it. And so scientists a few years ago figured out how to manipulate this system so that we could, in fact, use CRISPR in order to take genes out and add genes into different animals. And so there's been a couple of pretty cool applications to this that are still very much, in testing, but someday could be used more readily. So we talked about sickle cell anemia a little while ago. We talked about how that comes from a genetic mutation where a person has one letter versus another letter in their hemoglobin gene. And what then happens is that their red blood cells, when they're making that hemoglobin gene that has that mutation in it, it causes those red blood cells to have a sickle shape, to not have a round shape. So there is a group of scientists that are right now looking at trying to cure sickle cell anemia using CRISPR gene editing. And what they essentially are doing is they're starting with this one patient, and they took parts or they took cells from this person that has sickle cell anemia, and they used CRISPR in order to add the non-mutated version of the hemoglobin gene into them. And then they insert those cells back into the person to see if those cells will then start making the red blood cells of the round shape rather than the sickle shape. And so far it looks pretty good. It's not completely conclusive whether or not it worked, but this study started back in the fall, around November. And so far it seems like it's working. It seems like the person is in fact cured of sickle cell anemia. More recently, there's been an application with one type of degenerative blindness where a person's retina starts to degrade. The cells that are inside of the retina degrade and it causes them to lose vision. So there was a very recent test that was done just this last week or two in which they used CRISPR and inserted viruses with CRISPR with the correct DNA to correct that degenerative blindness. They inserted that into this person, into their eyes, and now they're looking to see whether or not their vision does improve. So instead of actually taking cells out of that person, they just used cells with the CRISPR technology in it and put those into the person. So that's still very much early in its development. We'll see whether or not it worked or not. But that is another kind of interesting application. And all this kind of gets at how CRISPR is a really, really powerful tool and could be a very powerful tool in medicine, in years to come. Because it allows you to do things in a much more precise way than you could in the past. Right, so in the past, there would never be a way that you could just take all the red blood cells inside of a human that have a sickle shape and replace them with a circle shape, because that person's body is going to just keep making more cells with the sickle shape. But if you can modify their DNA so that now their cells are making normal round shaped cells, then you essentially have cured sickle cell anemia. So this is going to be something that you should keep paying attention to, because we're going to see throughout our lifetimes more and more use of CRISPR for curing various types of diseases. Okay, so that's all I've got to say about DNA technology. In the next chapter, we're going to kind of shift to something else. We're going to start talking about how cells divide, and we're going to get into genetics. So that's all for now.